Hi guys and welcome to Mindset Learn. It is spring school. This may be the last session, but don't you worry because there are repeats on this entire weekend. And as you can see, I've got Eugen standing next to me. How are you?
Good and yourself, Indy. How are you doing this afternoon?
So good. I'm very excited for today's show. I think it's going to be lots of fun and lots of interacting.
Very interesting topic we're doing today. And I'm sure we're going to get lots of interaction from the page.
Yeah,
Reproduction, guys.
Reproduction, exactly. And I'm looking forward to all of those questions, guys. Keep them coming, and let's get. We're not going to get Eugen blushing here because you've seen all of the questions before. I, on the other hand, might blush. I might blush.
But I suppose you guys have been through this, so let's have a look at some of the questions.
That's exactly what it is, guys. This is serious learning. Do you want to take a spot by the desk?
Thanks, Indy.
Okay, this is what we're going to do, guys. We're going to get chatting. Please join us on facebook.com/learnextra. I'm also here for you guys on Twitter @learnextra. Please make sure that you post any questions as we go along. We'll be posting our challenge question. It's coming up next, so you better be keeping your eyes peeled for that. And if you don't know by now, there are many a way to win this awesome prize. What you need to do is you need to find the challenge question on our Facebook page, and please post the code as well as the correct answer underneath there, so you could possibly win one of these this awesome prize. And in this prize is this awesome CD, which is the Platinum Edition, Mikasa. We've got Electro Candy 4, which is actually 3 CDs in one. We've got this awesome textbook, which is a life science, which you could win too. And don't forget to get out your spring school books, guys. This is what you can actually work through with us. If you don't have this, I will be posting a link on Facebook so you can download it for free. We've also got this awesome t-shirt, so we're giving away. And just for today, because this is kind of like our last show, excluding the repeats this weekend, we are giving away this DVD, and it's Rock the Mother Tongue. And what it is, it's an award-winning comedian of 2012, so that can get you laughing. And you can watch this after matric and laugh all the way. So what we're going to do is we're going to get started. Big up to Liberty. Thank you for making the show possible. And Eugen, I can see he's like, come on, I want to start the show now. So get chatting to me. Eugen, take it away.
Thanks, Indy. Guys, awesome prizes up for grabs. I actually think I should take away those prizes. But anyway, I think I know this answer, and I'm going to leave it to you guys to look at the question. So let's look at this question. So the question for this afternoon is, what process does the term oogenesis refer to? We've got a few options here, and let's look at them. The options A is the secretion of estrogen, B, the production of haploid egg cells in the follicles of the ovary, and C, the production of the male gametes in the testes. And if you guys remember, your teachers would have done this with you. This comes under the importance process where we talk of gametogenesis when gametes form. So no clues further than this. Let's see what answers you guys are going to post. So from my side, Indy, let's look at those answers, and I'm sure there's going to be lots of correct answers. So we're going to look at today some questions on reproduction. I'm going to go through many of these questions. I want to see you guys posting some questions on the page. And it's about that time when you guys have gone through your prelims. You would have identified areas that you're having a difficulty with. So let me have those questions, and I'm sure we're going to be able to solve them today. So let's look at our first question today. Okay, so this was a question that was taken up from one of the tests that I set for some of the learners at the school that I teach. Let's read this question. The secretion of LH is controlled by LH-releasing factor, a small peptide molecule in the hypothalamus. LRF can be manufactured in the lab, and it's known as synthetic LRF. The graph below shows the effects of synthetic LRF on LH secretions in a healthy woman. LRF was injected on day 3 and on day 11 of the menstrual cycle. The same dose was injected on each occasion. Let's look at this graph. When we look at this graph, on the x-axis we can see there's time period, and on the y-axis we see the levels of LH in the bloodstream. It's very important for us to be able to understand the graph before we attempt to solve these questions. So the graph on day 11, I'm going to just highlight that in the yellow for you so we can clearly differentiate between the two graphs. I'm going to change my pen. I'm going to use another color for the graph that represents day 3. So that is day 3, and that's the dose of LRF on day 11. Very interesting information on this. Okay, So we clearly see, if we interpret this graph, we can clearly see this is the time 0. This is the levels of LRF, the levels of LH before the injection of LRF injection, and this is 3 hours after the injection of LRF. And before we attempt to answer these questions, I just want to quickly chat to you guys about LH and FSH. If you guys remember, the pituitary gland or the master gland produces two hormones, FSH and LH. FSH stands for follicle stimulating hormone, and that stimulates the development of the follicle in the ovary. LH is released by the pituitary gland, and that controls the ovulation as well as the development of the corpus luteum. So let's look at this question now. Obviously, this is related to the graph, and the question says, comment on the difference in response to LRF on day 3 and on day 11. So we've got a comment, and this question is out of 2 marks. It's very important for us to look at the mark allocations when we attempt a question. So we've got a comment on the difference in the responses between day 3 and day 11. When we look at this graph, we can clearly see that there is a substantial difference in the levels of LH both on day 3 and on day 11. So let's try and understand why is this so. Okay, so when we look at the injection on day 3, we can clearly see here, let me change this, we can clearly see an increase and then a decrease, and then it remained very low. In comparison, when we look at the dose on day 11, we can see it did increase. However, when it decreased, it remained relatively higher than on day 3. So I would respond to this question by saying that when the dose was administered on day 3, the levels initially increased and then decreased to about 10 units per cm cubed. You can clearly see that about 10 units on day 3, as compared to LRF injection on day 11, where it initially increased and then it got back to a level of about 50 or 45. So you'll see that the effect of the hormone was different on these 3 days. Initially it increased on day 3 and it got back to a level, and then it further decreased to about 4.5, and then on day 11 it increased and it only decreased till about, and the value for that was about 45. Guys, it's very important when you have a question like this that you use the units from your graph and you use those in your answers. The examiner wants to see that you're able to interpret the information in the graph and present that to write an answer. Let's move on to the next question. Suggest a possible medical reason for the application of synthetic LRF. Now, if we guys remember what the function of luteinizing hormone was. Luteinizing hormone was to maintain the level of, to stimulate the production of progesterone from the corpus luteum, as well as to stimulate the initiation or the process of ovulation. So when we look at LH stimulates ovulation. And if you guys remember, ovulation takes place on day, and that is on day 14. On the 14th day. So the question is, suggest a possible reason why the application of synthetic LRF was needed. Okay, simply, if you were a doctor and you had prescribed synthetic LRF, you want to stimulate ovulation. So the answer for this would be to stimulate ovulation. And that's exactly what it was given for. It was given so that ovulation could be stimulated. And often this happens in females that do not ovulate at their proper date in their cycles, and it's also to stimulate the release of an egg so that fertilization could take place. That was a 2-mark question. Let's move on to the next question. Okay. Still related to the graph. And obviously this is a question that I just answered now. State one function of luteinizing hormone. And as I said, luteinizing hormone, if we go back to the pituitary gland, I'm very quickly going to do this for the benefit of those of you that cannot remember. So we have the pituitary gland that produces LH, and LH would be targeting, this would be the Graafian follicle. Let me change that, guys, to show you the corpus luteum. Okay, and let me show you the rest of the, okay. So what I'm trying to draw here is the oviduct, the fallopian tube, and show you the ovary. And that ovary will release a secondary oocyte. I'm going to use lots of colors today so that you can clearly see what I'm talking about. That secondary oocyte will be released, and that will then travel into the fallopian tube. Okay, so this structure that is releasing the secondary oocyte is the Graafian follicle. And it's very important that you remember that. And that follicle will only release the secondary oocyte, on day 14, and that's ovulation on day 14. So LH is going to target the corpus luteum, the Graafian follicle, to release the secondary oocyte. And that secondary oocyte will then travel up into the fallopian tube for fertilization to take place in that area. Okay. So on what day, okay, let's go back to that question again. We said one function of luteinizing hormone, and that would be it is responsible for ovulation. Okay. And it also, that's one of the answers. The second answer is that it stimulates the corpus luteum to produce progesterone. Okay. Let's move on to the next question. The next question is again related to that. On what day of the 28-day menstrual cycle are the circulating levels of LH the highest? I've answered this question as well. So remember that ovulation takes place on day 14. So if ovulation takes place on day 14, the levels of LH would be obviously high just before, at the end of day 13 and on day 14. So the levels of LH will be the highest on day 14. Okay, so it'll be on day 14. What I'd like to do is very, very quickly show a little graphical representation of the hormonal levels during the menstrual cycle. So I'm going to draw a very simple graph here. So I'm going to extend the page for you guys. I'm going to show you an x-axis, and that is time, and that's day 1 to day 28, and that's on day 14. I'm going to just show you two hormones. I'm going to use yellow to represent day 14, and that would be LH. And I'll show you FSH. So that's we're calling LH here. And this would be, I'll use another color, FSH. And this would be FSH. And from this graph, you can clearly see, that on day 14, when ovulation takes place, the levels of LH would increase and then gradually decrease. Body's trying to be funny with me. Let's try that again. Okay, never mind. But I hope you
click and move.
Okay.
Never that the board is misbehaving today.
Okay, but anyway, we can still see exactly how this level would increase. Okay, let's move on to the next question. Okay. This question also relates to the use of infertility treatment for women that find it difficult to conceive or that find it difficult to have a baby. Okay, so in some women, infertility may be treated with clomiphene. Clomiphene is prescribed to certain women that find it difficult to fall pregnant, so it will help in the stimulation. It will stimulate the ovulation as well as the production of eggs. So let's read further and highlight the key points in this. Clomiphene stimulates the production of gonadotrophin from the anterior pituitary gland. So this will stimulate the pituitary gland to produce gonadotrophin, and that will then stimulate the pituitary gland to produce more FSH, and that will mean that there's more follicles that are produced in the ovaries. The graph below shows the blood estrogen levels in women. So it's the estrogen levels that in women during and after the treatment with clomiphene. Again, and these are the types of questions that you're going to get. You're going to get the interpretation of information presented in a graph. And it's not difficult. You've got to be clear and understand how the information is presented. Look at the x-axis, look at the y-axis, and try and understand the graph before you try and answer those questions. So that's exactly what I'm going to do now. So I'm going to look at what's on this graph. The graph shows me time on the x-axis, and on the y-axis we see blood estrogen levels, and that's measured in micrograms. So we see this over a period of from day 1 right up to day 26. So obviously this is showing us the levels of estrogen over a period of 28 days, or in this case 26 days. And here we clearly see, looks like the Manhattan, but we can clearly see an increase, then a decrease, and then an increase again, and then a gradual decrease. And what's important to remember is that these changes are cyclic events that take place throughout the cycle. So let's look at some of the questions. From the timing of the treatment shown in the graph, name the gonadotrophic hormone whose secretion is stimulated by clomiphene. Now, this is a 2-mark question, guys. And some of you may say, well, 2 marks, probably very easy to answer this, but let's look at that question again. It says, let's name the gonadotrophic hormone. Now, when you see the word gonadotrophic hormone, it refers to the gonads. So this will obviously refer to the gonad or a gonad that produces a hormone. So let's not rush into the question and assume the obvious. We've got to read carefully. So we're naming a gonadotrophic hormone whose secretion is stimulated by clomiphene. When we look at this graph, guys, we can see if we think about the gonads and the females, the female gonads or the ovaries produce 2 hormones, and they produce estrogen. Any guesses, Indy, which the other one would be?
Gosh, it produces estrogen.
And the other one?
Progesterone.
Excellent.
Thank goodness that was right.
Right. And this is the reason why we have progesterone. Let me write that down. Progesterone. And I'm going to ask, okay, so, and I often ask my boys that I teach, or the learners that I teach, in terms of how do you remember the functions of these hormones? And I use a very easy way. I use the P in progesterone to say it's got some function association with pregnancy, so that obviously maintains pregnancy. And estrogen, the O in estrogen would be related to ovulation. Okay, so estrogen levels are associated with ovulation. So name the gonadotrophic hormone whose secretion is stimulated by clomiphene. And actually this is right here in the graph. So it's estrogen. Okay, so we looked at that. So let's look at the next question. From which organ is estrogen secreted? Well, guys, I've answered that question for you. Clearly, I showed you here. The gonads that secrete estrogen are the ovaries. And that's only one of the hormones. The other hormone is progesterone. Let's move on to the next question.
Jürgen, you ready for a break?
I'm sure. I'm actually looking forward to that
he's
break.
he's enjoying it so much. You just carry on the entire time. But I need a break. You guys need a break. It's so hot today. Go and get yourself something to drink. Make sure it's water, because it's good to make sure that you are really being able to pay attention. I do want to read out a really inspirational message from a mindsetter, which I thought that was really, really cool. And she said, guys, be the change you want to see in the world. Have a positive attitude and you will succeed. I couldn't have said it better myself. That was awesome.
Awesome.
Yeah, very, very cool, guys. So what we're going to do is we're going to take a little break, and when we get back, we will learn more and learn extra. Hi guys and welcome back. We are having such an awesome lesson and we've been looking through the page and finding those questions that you've been sending in, and I think we've actually got one from Jarnik which I'm going to read out and we're going to work through. But before we do, I know Tusa was saying to me on the page, he was saying he's so sad that today is the last day of spring school. No guys, no. Maybe the last day of spring school, but there are repeats this entire weekend. And not only that, we've got exam school starting too. And how that works is 1 or 2 days before you write your finals, and it corresponds to the final timetable, we will be having live shows, so you will be able to get that one last push in, that one last little lesson that you need just before you write the finals. And we'll be spending the entire month with you and we're going to be with you during finals. We are here for you for the entire journey. So let's get going. So here's Jarnik's question. Are you ready?
Right, let's have the question, Indy.
Okay, so this is the question. She said, "Hi, Indy. I just wanted to know the function of the Sertoli cells and of the Leydig
cells.
Thank you again.
Sounds very nice when you say it.
Oh, does it? It sounds a little bit like foreign. What's the difference? Got confused in the last section when they said the cells of Sertoli secretes testosterone. Don't they have glycogen that serves as nutrients for the spermatids?" That is the question.
Okay, there's some misconceptions in the thinking there, but I'm going to very quickly clear up that. In fact, by coincidence, the next question actually deals with the cells of Sertoli. But I'm going to answer your question first, and then we're going to look at what I've said for you this afternoon. So there's two cells that we're talking about. Let me just change my pen. So the Sertoli cells and the Leydig cells. And guys, it's obviously scientists get acknowledged for their work, and obviously these are named after people that were involved in identifying them and understanding their function. So let's see. So remember that these cells are, when we look at the seminiferous tubule, so if I were to draw a diagram of the testes, and we've got the epididymis on the surface of the testes, and that goes out into the vast sperm duct. If we take a cross section into that and we look at the cells inside the seminiferous tubules, we've got a layer of germinal cells here that produce spermatids. Okay, we've got also developing sperms that grow within the seminiferous tubules. Okay, so the two cells that we're talking about today play a very important role. Okay, and I often use the S in Sertoli for the word nurse. So you guys will know what nurses do. They tend to look after you, and they tend to help you when you're growing, when you're sick, and when you're growing up. So that's exactly what the cells of Sertoli do. They will nurse the developing spermatids so that they mature and they grow. So the function of the cells of Sertoli is to nurse this developing cells of spermatids. On the other hand, we have the Leydig cells. It's also called the interstitial cells. Interstitial cells. And the word interstitial cells obviously means cells that are found in between. So these cells are found in between, and they play a very important role in, yes, producing the male hormone testosterone. So the testosterone is produced by the Leydig cells, and they help and stimulate the development of sperms, and these will be then stimulated by the hormone LH. So LH stimulates the Leydig cells and the interstitial cells to produce the male hormone testosterone, which actually help in the development and maturation of the sperm. So that's exactly, and I hope this answer answers your question and clears your doubt about, yes, the fructose that you're talking about and the energy that is needed for the development of the sperm comes from the cells of Sertoli. Also, during the secretions from the different ducts in the male reproductive system, you'll find that secretions are secreted from the seminal vesicles that actually nourish the developing sperm as it matures and completes its development in the sperm duct. Let's move on to the questions that I'm just going to clear this very quickly for you. And I'm glad you asked those questions, and that's what you should be doing now, guys. You should be posting as many questions, because the doubts that you have can be cleared up now. And the ideal thing is to ask as many questions as you can, so we can iron out all those confusions that you have. And this is the best time to do it. So let's not despair about it being the last lesson. Let me have your questions, and I'll try and answer as many as I can. Let's look at what I've presented now. Okay, so here, when a male contracts mumps, Okay, this is a viral infection, okay, which affects the glands in the neck. There is usually a fear that the virus will spread to the testes. The virus will cause the damage to the cells of the Sertoli. So you'll find that kids often, when they're young, they pick up infections, and one infection is mumps, and it does affect the lymph nodes, and sometimes that can also can spread into the gonads and the genital areas, and there is a concern that if it does, that it could affect, and it does target and affects the Sertoli cells. So if it affects the Sertoli cells, then there's concern about the development of the sperm and the fertility of the young boy as he will grow up. So let's look at the question. So here we see a layer of connective tissue with the Leydig cells, and as I just mentioned, the functions of the Leydig cells now. Let's look at the first question in this. So here we see a Sertoli cell, and if you just remember from what I said, I also call it the nurse cell. And well, you're not going to get a mark for saying it's a nurse cell, but I use the word nurse so that I remember the function of these cells are to nurse the sperm. And if you were to use that in your explanation for the answer, to nurse the cells, and I'm sure your examiner, the marker will give you the benefit of the doubt, but they're not called the nurse cells. They're used to nurse the cells in their development, the young sperms in their development and maturation. So let's look at the first question. What would the implications of testicular mumps be on a male? Now, if we looked at the function, the question tells you that it will cause damage to the cells of Sertoli. If the cells of Sertoli are damaged, guys, what is more likely to happen? Yes, if you look at it, if these cells of Sertoli were damaged, what's going to happen? These young cells, the young spermatids, are not going to have nourishment. They're not going to be nursed. They're not going to mature. So we're going to find that there's two things so we're going to find that what will happen, so he could become sterile, which is not a good thing. That means you will not be able to produce enough sperms, or your sperm count will be low, or we could also say it could lead to infertility, which is another word, a fancy word for saying will not be able to produce enough of sperms. Okay, and that's a 2-mark question. Let's look at the next question. Describe the function of the cells of Sertoli. And I just did that, guys. So if we were to describe the function of the cells of Sertoli, I'll repeat that again. It's there to produce it. It's there to nourish and to nurse the developing spermatids in the seminiferous tubules. Okay. Right. Let's look at the next question. Study the diagram below and answer the questions that follow. And you guys would have seen diagrams like this before. And remember, I said whenever you see a diagram in your question paper, do not scramble to read the question and try and attempt to answer the question. Try and look at the labels, the label lines or the parts that have been labeled. Very quickly write on your question paper the functions or the parts of those to those labels, so that when you answer the questions, you can clearly, you don't have to go back and think about the answers. You've got them, and often that helps in terms of moving through the question very quickly. So let's look at this. So I'm going to very quickly label this diagram for you. So the diagram obviously is of yes, it's a male sperm, so it's a sperm, so it's a diagram of a sperm. I'm going to label that for you very quickly. So the sperm can be divided into 3 sections. So that's the head, the mid-piece, the middle piece, and some of us tend to call it the body. And we obviously have a long tail. Okay. And we've got part labeled F. F would be the acrosome, and that's found in the head. We can do the functions of these in a little while. E would be the nucleus, which contains the genetic information. D would be, if you guys remember from grade 10, that's the mitochondria. And we'll go into the functions in a little while. Okay, so the first thing that we need to do is the first question says, identify the structure. And as we did that now, that's the structure of a sperm. That's the male gamete. That's the gamete. I'm going to very quickly ask you guys, and I'm going to give you another clue as to the question that was posted, the competition question that was posted. So the formation of sperms takes place through gametogenesis, and male sperms, or rather sperms, are produced through a process called spermatogenesis. So you guys got to now remember the question, spermatogenesis, and use this as a clue to figuring out what oogenesis was. Okay, let's move on to the next question, guys. Still relating to the diagram. Identify the parts labeled C, and we just did that. So C is the mitochondria or chondrion, singular. And D would be, sorry, I just got that wrong, guys. Let me just, that was actually, okay, let's do this again. So C was the tail, and D was the mitochondria. Okay. Let's move on to the next question. Right. What is the chromosome number of the part labeled E in a human? And that's a 1-mark question. So if we look at part labeled E is the nucleus. So we want to know what is the chromosome number of the nucleus. And guys, if you remember, I spoke about the process of gametogenesis. Gametogenesis is the formation of sperms. And if you go back towards the start of the year when you guys did cell division. So cell division there are 2 types of cell division, mitosis and meiotic cell division. Gametogenesis is a type of meiotic cell division in which sperms form. Now, if you recall, meiotic division would mean that there is a reduction in the chromosome number. So if sperms are formed through a process known as gametogenesis, what would the chromosome number be of the nucleus in a sperm? Yes, it would be half the full set of chromosomes. And in the case of humans, we have 46 chromosomes. We have 46 chromosomes. And I'm just going to, so we have 44 autosomes, guys. And autosomes, I'll explain to you that right now. And we have 2 gonosomes. I've introduced 2 new concepts. You've probably heard these terms before. So somes obviously refer to the chromosomes. We have 46 chromosomes in a normal cell. We have 44 autosomes, as you clearly see here. And we have 2 gonosomes. So the gonosomes are the sex chromosomes, or the chromosomes that are associated with the determination of the sex. And your autosomes are those chromosomes that are normally associated with your genetic information responsible for all other functions except the determination of sex of an individual. So in the nucleus, we will always have, I'm going to use the term haploid. And you've heard of this before. We would have haploid number of chromosomes. And this simply means that it is half the full set of chromosomes. And if we take 46, guys, and divide that by 2, so it will be 23 chromosomes. We want not to make the mistake 23 pairs. 23 pairs again would mean that it's 46. So it's 23 chromosomes because that cell has been produced because of a reduction division or meiotic division. Okay. Right, let's move on to the next question. Still busy with the sperm. What is the function of part labeled F? Guys, if you remember part labeled F, we labeled part F in the previous page. It was the acrosome, and that is found in the head of the sperm. Okay, so I'm going to very quickly draw you an egg cell, and I'm going to show you a sperm that is swimming along, happy, reaches the egg, and says, knock, knock, let's enter. It's not going to be able to enter without being able to penetrate the wall of the egg. So the egg needs to be penetrated. In the head of we have the sperm, we have the acrosome. And the acrosome actually produces an enzyme. And that enzyme plays a very important role in digesting the membrane of the egg so that the sperm can penetrate the egg and enter the egg so that fertilization could take place. So the function of the acrosome would be to penetrate the egg, release an enzyme, and enter the egg. So guys, we've done a lot. I think it's time for you guys to have a break. I'm going to hand you over to Indy, and then I'll see you around.
That's exactly what we're going to do, guys. We're going to take a short little break. Go and make sure that you do get yourself a glass of water because, sure, it is hot. It's even hot here in studio. Calvin says the future belongs to those who believe in the beauty of their dreams. If you fail today, dust yourself off and say, I will pass tomorrow. But, guys, you're all going to pass with flying colors. Make sure you post those questions onto our page. Like we always say, there's no such thing as a silly question. Only the question that is asked after the exam because then it's too late. Get those questions in, and we'll see you now after the break. Hi guys, and welcome back to Spring School, and we are on life science, going over reproduction. So I hope that you are tuning in and you are getting those questions in on the page. That's facebook.com/learnextra. Also chat to me, Indy, @learnextra on Twitter. Guys, we are having such an awesome time, and you are posting amazing questions on the page as well as amazing inspiration. Rudolph says the longer you're not watching Learn Extra, the more marks you're losing. So thank you for that. I thought it was a very cool comment. Guys, I want to say a big up to Liberty. Thank you guys for making this show possible. And a quick little recap of what you can win. 3 CDs and I think it's an award-winning Comedian of the Year 2012 DVD, an awesome T-shirt, as well as a life science textbook. So make sure that you do check out the challenge question. Don't forget the code, and we will be closing that, I think, pretty soon, but we'll let you know when we do. And I think what we're going to do is we're going to go straight over to Jürgen. Let's not take any more away from your lesson. Keep those questions coming. Jürgen, take it away.
Thanks, Indy. Guys, don't forget, I've given you at least 2 clues already to answering that question. I know those prizes are fantastic, so let's get those answers in before it shuts down. So let's continue with the question that we were busy with. So just before we went on to break, I described the function of the acrosome, which is found in the head, and that produces an enzyme, which actually will digest the membrane around the egg so that the sperm can enter. And once it enters, the egg will produce an enzyme that prevents any of the other sperms from penetrating and thus preventing double fertilization. So let's move on to the next question. And one similarity between this cell and a pollen grain. Now, I'm going to just extend this, and it doesn't ask us to tabulate, but I'm going to do this in a table for you. So we're talking about a sperm cell and a pollen. And guys, you got to remember, you cannot compartmentalize your whatever you study. I mean, when you study plant reproduction and you study human reproduction, do not expect questions to be asked exclusively on plant or human reproduction. Remember that the examiner wants to see how you can integrate your understanding of the concepts. And hence, this is a question that brings in 2 different topics into one question. And you've got to look out for that. And remember that you've got to apply what you've learned in reproduction, in plant reproduction, in human reproduction, and in cell division to answer all your questions. So do not get caught into thinking that this question is exclusively on reproduction. The examiner will bring in questions that are from reproduction, from cell division, from plant reproduction, so that he could test and see if there's a thorough understanding of knowledge. So let's look at this question again. So let's look at the similarities between the sperm and the pollen grain. So if we look at the sperm, guys, I'm going to put down the sperm is basically N and N. By N, I mean it is haploid. So both cells are haploid, so they have half the full set of chromosomes. So sperms and pollen are haploid. We can also say that they are gametes, so that they are, by gametes I mean that they are reproductive cells that are produced by, both are produced by cell division, and the type of cell division that is produced is by meiosis. So we could come up with several produced by meiosis. Okay, and they both play an important role in fertilization. So they're involved in fertilization, and they are male gametes. We mentioned that already, but both are male gametes. Okay, so a question like this would normally come for, in this case, because it's one similarity, it's a single mark question, and hence you've got to give at least one similarity. However, if this was asked for 3 marks, you would then need to give at least 3 similarities between the two cells. So let's move on to the next question. Okay, so we've done with that question. We've gone through what is the chromosome number of the right. So we're coming to questions that have been taken out from the Feb-March exams from paper 2, and it's 2012, so the exams that were written in March this year. So let's look at them. These are multiple choice, and obviously, guys, we tend to rush into the multiple choice, but please read those questions, read the options very carefully. There's never an obvious answer. There's always an alternative that's very close. So read, eliminate the obvious, and then you'll find the correct answer. So let's look at the question. The question goes, which one of the following refers to the development of some birds where the egg hatches outside the body and the young are born immobile and totally dependent on its parents? Now, this takes us to, and I know that there was somebody that posted a question, and the question that they posted was on, and Indy was having a tongue twister with viviparity, oviparous, and I'm going to ask her to throw that question again. Let's have a go at her for the last program this season.
Jomi, shall I read it out
Please.
to you? Oh, okay, here we go. Okay, Monty has asked, what is the difference between oviparous and oviviviparous? There is, and what I think John said that there is a question coming up after the break, and I think we're about to go after this question now.
So let's look at this. So I'm going to read this question. Obviously, that's going to help you to understand the difference between oviparous, viviparous, and ovoviviparous.
Ovoviviparous.
Okay.
I will get it by the end of the show.
You will definitely get it. Okay. So the options are viviparity and precocial development, oviparity and altricial development, viviparity and altricial development, and oviparity and precocial development. Now, we've got to look at the question again and try and understand exactly what we're trying to answer here. So some birds lay eggs, so eggs are laid, and they hatch outside the body. When we look at reproduction, guys, I'm going to very quickly I'm going to extend this page for you. So we have eggs that are laid, and the offspring are born. In these eggs, we have live births. So we have eggs that hatch. We have organisms that give live birth to live births. An example would be humans. So where we find individuals undergo gestation. And these individuals will carry the young through a gestational period, and that develops inside the uterus or the womb of the mother, whereas in organisms that lay eggs, the egg will hatch outside the body. So this is external development. Okay. And then we also get organisms that produce eggs, and the eggs are incubated internally. So eggs are laid and they are incubated internally. And we often see this, we see this in sharks as well. Some of the sharks, some fish tend to do this. Some fish, not all fish, some fish tend to do this. So we haven't answered the question yet, but I'm trying to categorize 3 different terms. So we have the terms, so we refer to humans as viviparous, and that's viviparity. These organisms as oviparous, and these as ovoviviparous. Okay, so that's your 3 broad categories of organisms based on the ability for gestation and fertilization. So you'll find that fertilization happens externally. There's internal fertilization with viviparous, and in oviparous there's external fertilization, but the eggs will undergo development within the body of the mother. Let's look at the question now. Okay, we're going back right on to the top. So, which one of the following refers to the development of some birds where the eggs hatch outside the body and the young are born immobile and totally dependent on its parents? So when we went back, so it's definitely oviparity, So there's eggs that are laid, and there's some amount of parental care. So when there's parental care, we call precocial development, and that would be the answer, where the parents offer parental care and nurturing to the young ones. Let's move on to the next question. The statement below refers to the action of different contraceptive methods. Okay, so we've got these 4 statements, and we've got to respond to the question. The question is, which combination refers to the action of oral contraceptive pills only? So let's look at these statements and try and see what they talk about. Statement number 1, it says inhibits the secretion of FSH, so FSH is inhibited, increases the levels of hormone progesterone, stops the embryo from implanting in the uterus, and stops ovulation by inhibiting the development of the follicle. Now, if we were to look at these options, these are typical statements that relate to how the different contraceptive methods function. So if we look at contraceptive method broadly, there are 3 major categories, or rather 4. If we look at chemical, which would be the administration of the pills and the patches, or even through an injection. A mechanical barrier would be through the use of barriers such as condoms, femidoms, diaphragms that prevent the sperm from entering or prevent fertilization. And we also have the third, which is a surgical procedure, which often requires which would be ligation or a vasectomy, which would mean that the sperm ducts or the oviducts are cut up and knotted so that the ovary does not reach the eggs do not reach the uterus, or the sperm is not released along with semen. So let's look at this question. So this would be inhibits the secretion of FSH will be the pill. Increases the level of hormone in progesterone, again, that will be the pill. Stops the embryo from implanting in the uterus. That would be either the IUDs, the intrauterine devices, so that could be a copper loop or an IUD. Stops the ovulation by inhibiting the development of the follicle, and that would also be the contraceptive pill. So if we look at options here, so it's definitely 1, 2, and 4. We say it's not 3, and the options here 1, 2, and 4. So the answer for that would be B. Okay, let's move on to the next question. Which one of the following is true about seeds? Now, remember that plants produce seeds as a reproductive structure, and that seed is capable of growing to a new plant. So let's look at the question. So the functions of the seed which one of the following is true about a seed? So it's not necessarily the function, but true. So it protects the gametes, provides the embryo with food for the cotyledons, yes. Development into a fruit, does not do that. Develops to form an ovary, and that's not true. So the answer for that would be B. It provides the embryo with food from the cotyledons, and that's essentially what the fruit has. So in the middle, when you eat a fruit, you find that the seeds are there, and we tend to eat the fruit, the fleshy outer parts, that's the fleshy endosperm, and the seeds is often the parts that we don't eat. And that seed actually, if you look at a typical bean, guys, you'd see that when a bean sprouts, the cotyledons are the part of the fruit that actually provides the energy for that developing seed to grow into a new plant. And I know we're almost due for a break. I'm going to do this question, and then we're going to go into a quick break. Do we have time for this question, Indy?
Yes, we do.
Okay.
Just do it. Yeah,
Let's
definitely.
do it. Okay. The statement below refers to the adaptations of flowers for pollination. So pollination is a process where the pollen from the anther is transferred onto the stigma. So there's 2 types of pollination: self-pollination and cross-pollination. So let's look at this question very quickly. So how are plants adapted for pollination? So the male and female flowers are found in different plants. The stigma below the anthers, the male and female gametes mature at the same time. Let's look at which combination of these refer to self-pollination. Okay, so self-pollination again is when, guys, very quickly, the anther produces pollen and that lands on the stigma. And this has to be on the same flower. So let's look at the male and female flowers are found in different plants. That would not be true, because for self-pollination, they need to be on the same plant. And here it talks of male and female flowers, so that's not self-pollination. Stigma below, the anthers, and here you can see that the stigma is there to receive the pollen, and male and female gametes mature at the same time, and obviously so that self-pollination can occur. So the most correct answer for this would be, we look at number 1 is obviously not correct, number 2 and 3. So the correct answer for this would be the option C, which is 2 and 3 only. And that is essentially how self-pollination would occur. Guys, I think we need a break. It's been a long session. I'm going to hand you over to Indy, and we're going to go for a short break.
Okay, guys, if you haven't put in your answer to the competition question yet, please do so now. I'm sure that it will be closing in the next few minutes, so just make sure you do that. Just to add to what Calvin said earlier, and this is from Nakedi, he says, champions try and fail, but they never fail to try. Thought that was awesome, and we'll see you now after the break. Hi guys, and welcome back to the show. We are having such an awesome time, and keep on posting onto our page. There's no such thing as a silly question, only the question asked after exams. And I'm so proud of you guys because you all help each other out on this Facebook page, and that's what this is all about. This is about not just us having this page, it's about you having the page too. It's our little Mindset Learn community. So help each other out, guys. If you see somebody posting a question that you know the answer to, sometimes it's a nice way for you to learn as well by helping out and posting the correct answer back. So now I think what we're going to do is we're going to go straight back to Jürgen, and then we've got a few questions that we need to be answered, but keep them coming. Facebook.com/Learn Extra, and also chat to us on Twitter @Learn Extra, and I'm bumbling away here. So I think, Jürgen, it's time for you to take it away.
Thanks, Indy. Some interesting questions, guys. I had a look at some of those questions. We're definitely going to get to those questions, and I looked at those questions. Some of them relate to the questions that I'm doing now. So whilst I'm going to be busy with this, I'm going to ask Indy to throw me those questions so that I can integrate your questions with these diagrams, and it'll definitely make more sense to you. So let's look at this question. So the next question obviously deals with pregnancy and the period of the gestational period up until labor and post-delivery. So let's look at this. The diagram below represents the events leading to the development of the fetus in the human uterus. So we've got a nice diagram here showing the parts of a female reproductive system, showing various stages in the development of the fetus. And the stages that fetal development is, that term is referred to as the gestational period. So if you saw the word gestational period, it refers to the period of development of the of the baby or the fetus inside the uterus or the womb of the mother. So here we see a diagram showing you a cute little babaki here that's developing. Here you see the ovaries, and here you see the fallopian tube, and a series of events happening in there. Okay, so let's look at this question. And as I said, it's important for us to label these diagrams before we attempt to answer the question. So that's obviously the ovary. It shows you the ovary. Number 2 would be what is released from the structure here, guys. And we spoke about the Graafian follicle, and that's the secondary oocyte. And some of you tend to call it the ovum, but I still prefer using the secondary oocyte, which is the most correct description for that. Number 3 would be showing you the zygote. And we go to number 4, and we just did that structure there with a little tail, and that's the sperm. And if we look at number 5 shows you the developing fetus and the attachment of the fetus to the maternal sinuses, and that structure is the, yes guys, the umbilical cord. And number 6 points to the liquid inside there, and that's the amniotic fluid. Okay, so let's very quickly look at some of these questions. Okay, identify the following parts, and we've got to label part number 1, and we just did that now. Part number 1 was the ovary. Okay, let's look at the cell labeled 2. Here we've labeled cell, and we said that that is what is released on day 14 of ovulation, and we spoke about that. That's the secondary oocyte, guys. and it's from the development of the primary oocyte. So that's the primary oocyte that develops, and it's released as the secondary oocyte. So it's the secondary oocyte. And if you guys remember the question that was posed to you, that's the third clue there. Cell labeled 3, guys, and that's produced after fertilization. So that's the zygote. Structure labeled 4, guys, we did that as well. Structure labeled 4 is the sperm. Lots of repetition because these are questions taken out from different papers, and obviously we want to expose you to many questions, guys. Structure label 5, and we said that was the placenta. So sorry guys, I'm getting carried away now. It's not the placenta, it's the umbilical cord. And structure number 6 we said was the amniotic fluid, in which the baby will bathe inside the placenta. So let's move on to the next question. Okay, and this question relates to many of the questions that you guys have posted. So I'm going to ask Indy in a little while to give me those questions. But let me explain to you what this graph is all about. So here we're seeing the graph showing the hormonal changes during pregnancy. Okay, so it does seem a bit complicated, guys. And remember that if you see a graph like that, the best thing to do is make it colorful. And believe you me that the colors definitely make understanding so much easier. So I'm going to use some different colors in here so that I can clearly see trends in this graph. I'm going to use a nice blue, and that's Indy's favorite color, I think.
It is my favorite color. How did you know?
Awesome stuff. Okay, and then finally the last graph showing you there. So when we look at this graph, it shows you the process from day 0 or time 0 right up till week 50, and that's time in weeks, and this is 40 weeks, and that's 40 weeks is known as full trimester. And then you can see they divided into different periods in a period of 40 weeks. And normally delivery and labor occurs around week 38. That's when we refer to as a full trimester. When we look at this diagram, it shows you the graph. It shows you hormones in the blood. I'm going to change my pen, guys. It shows you hormones in the blood, and these are given in arbitrary units, but again, it's comparatively given so that we can compare. Here we see progesterone being produced from structure A, guys, and that is structure A, and it shows you from week 0 to about week 11 or week 12, and then we see the purple graph that I've labeled here, and progesterone from structure B. We see the levels of estrogen in yellow, and we see the levels of oxytocin, another hormone associated with labor and birth, and the hormone prolactin, which is released after week 33, and that increases gradually and remains high after birth, and it's associated with lactation. Now, if you remember, lactation is a process of where the mother produces milk from the mammary glands, and that is stimulated by the hormone prolactin. So prolactin will stimulate lactation. And if you look at this graph, it starts, so the mammary glands start preparing for lactation as you reach full trimester, the last trimester, so that when there is birth, the mammary glands are ready to produce milk for the baby to start suckling. Okay, so let's look at the first question. Identify the following structures A. So structure A in this case refers to progesterone from structure A. So in the body, in the female, in the ovaries, which are the structures that produces progesterone? Normally, it's produced from the ovary. And in the ovary, we have the corpus luteum that produces progesterone. However, after pregnancy, and you can clearly see here, that's the next question, identify the following structure B. So structure B also produces progesterone. And if you clearly look at what happens in this graph, during the first 11 weeks, the secretion of progesterone is from the corpus luteum in the ovaries. However, that eventually the corpus luteum degenerates after about 10 weeks, and the function of secreting progesterone is now the function of the the placenta. So the placenta will produce progesterone, and that placenta will produce progesterone throughout the period from about the 5th week right up till the end or the initiation or the start of labor. Indy, we're going to take that one question now. The question that was posted in terms of the levels of estrogen progesterone as we approach.
Okay, perfect. Because that one was from Mindsetter Tuso, and Tuso's always rocking on the page, which we think is awesome. And what he wants to know is, what is the significance of estrogen and progesterone dropping towards the end of a pregnancy?
Awesome question. If you look at, I've actually got the next question. Isn't that a good coincidence that we're going to do
look
that?
at that.
So
Tuso, he could see it.
Tuso is obviously
He's on the ball.
He's on the ball, and he knows which are the important questions. So, awesome question. Let's look at this. We've got to explain the significance, very similar to what Tuso has asked us, between the levels of estrogen and progesterone dropping towards the end of pregnancy. And if you see this graph again, guys, I'm just going to highlight just the drop towards the end. That's progesterone that's decreasing, and we also see estrogen levels that are also decreasing. So what is the significance of this? Now, if you remember, what is the function of progesterone and estrogen? If we look at this graph, we can clearly see that as pregnancy progresses, the levels of estrogen and progesterone are high. So definitely, they play an important role in maintaining pregnancy. So what do they maintain? Yes, as the fetus develops, the fetus establishes a contact between the uterine wall through the placenta. That placenta needs to be intact and held well into the endometrial lining along in the uterus. The attachment and the stability of that attachment is maintained by the hormones estrogen and progesterone. Hence, as we get towards labor and birth, the levels of estrogen progesterone drop. Why? Yes, that is because the endometrium will now tend to shred away. It's the start of labor. The attachment between the maternal sinus and the placenta needs to detach so that the process of birth can start, and hence the levels of estrogen and progesterone drop as the labor starts. What initiates labor, guys? If you see in this graph, we can clearly see here the oxytocin levels also increase, and that is at the time of labor. So the hormone oxytocin actually initiates labor. It will work on the abdominal muscles and the muscles around the pubic area so that the contraction of the uterus and the uterine walls start helping. And that's the reason why women that are pregnant and in labor undergo contractions, and that contraction is because of the hormone oxytocin. And when the levels of oxytocin increase, they work antagonistically to estrogen progesterone. So you clearly see a drastic drop in the levels of progesterone, estrogen, and that is the onset or the start of labor. Okay, and the reason why we've got the word labor, guys, if you remember, labor is hard work, and obviously that's the difficult part in pregnancy, labor. Okay, let's go on to the next question. Okay, Indy, do we have some more questions that we can take?
We do. I actually have a few more relating to pregnancy, so I think maybe it's the best to just get those done now. Oh, gosh, obviously my computer. Hold on, it's happening. Okay, this is a very important one from Justin. If I don't ask you this one, he's going to be very angry with me. So Justin Naidu asks, this is a question he got in his prelims. What is the function of the mucus which forms at the cervix during pregnancy?
Okay, I'm very quickly going to extend this page here, and I'm going to draw the cervix very quickly for you. Okay, and that's the vaginal tract and the cervix. And here we have the uterine wall, and we have the development of the baby. And okay, not that good, but it does look like a baby. So what happens? When a female falls pregnant, and remember that the cervix is here in that area, the baby is now developing in the placenta, and there's a placenta here, and there's amniotic fluid. There's a placenta that develops, and there's amniotic fluid in that placenta. We find that the cervix secretes mucus and forms a plug, and it's called the cervical plug, and that plug will prevent, and it's very important in preventing the entry of foreign substances. it plays an important role in protecting the developing fetus and preventing infection. It also prevents the initiation of labor, and that is exactly what happens. So as oxytocin is released, you'll find that the cervical plug will tend to be released, and that is what is termed as the water bag breaks, or there's water released, and it's only because the cervical plug will dissolve close towards labor, and that will release some of the contents in the placenta, which initiates the process of birth and labor.
Okay, so let's go to a little ad break, guys. And I do know that there are a few more questions on the page. We'll take a few more questions when we get back from the ad break, and the competition is also now closed. We'll be choosing that winner during the break, so keep your eyes peeled, keep stuck to your seats, and don't go anywhere. We'll be back with you now. Hi guys, and welcome back. We are nearly at the end of our show. We've got about 13 minutes left. I can't believe how fast it's gone, and that's why I don't want to waste any more time. But I do have to stress to you guys that all of the live shows that have been on this week will be rerun this weekend. All Paper 1 is going to be on Saturday. All Paper 2 is going to be on Sunday. I will post the schedule on Facebook. That's facebook.com/learnextra. Please, please, please make sure that if you did miss any of the shows this week, that you catch up this weekend. All of our schedules and the downloads are on our website, which is, just in case you have missed it, is www.mindset.co.za/learn/spring school. So go and check it out. We also have exam school, which is happening at the end of the month. And guys, how it is working is, let's say you're writing life science, we will make sure that we have the live life science show a day or two before you write your final. So we are with you all the way to the end. I will post a full schedule so you can make sure that you do attend those exam schools. This is the final countdown. We are nearly there, and you are so close to the end, guys, which is so exciting and so awesome too. So what I think we're going to do is, Jürgen, I think we're going to carry on with the lesson, and then we will be announcing who the winner of that challenge question is, and also letting you guys know what the correct answer was. So I think without further ado, Jürgen, take it away.
Thanks, Indy. So guys, I looked at some of the questions, and there was a general concern about what gametes were and what secondary oocyte is. Gametes refer to reproductive cells, so the sperms and the eggs are gametes, but the secondary oocyte, or the ovum, can also be called a gamete, as it is a reproductive cell produced by gametogenesis, and it's for the process of fertilization. So let's go back to the questions that we were busy with before we got into the break. So explain what will happen if structure A, well, we've done that, guys. Let's move on to the next one. Right. Okay. This is an experiment, and obviously these are questions based on the experiment. So let's read through them. A group of grade 12 learners carried out an investigation to determine the effect of gibberellin on the germination of seeds. Now obviously this is a plant reproduction question, but also related to reproduction. So hormones are produced by the plants also affect how seeds germinate and how they develop. So let's look at how they carried out the investigation. The following procedure was followed. A sample of hazelnut seeds was divided into two groups, so they were divided into two groups, okay, A and B. Group A was treated with gibberellin solution. Water was added to the seeds in group B. So we had a second group, B, which was treated with water. Both groups of seeds were allowed to germinate for 16 days, and that was the time period. So they were allowed to grow for 16 days. Excuse me. And the percentage of seeds germinated in the two groups was recorded. So they measured the percentage of seeds grown, and then they plotted that information in a graph. The results of the investigation are shown in the graph below. So when we look at this graph, first let's read the heading that tells us about the graph. So it shows us the percentage of germination of hazelnuts in gibberellin solution, which is A, group A, and water over time, which is group B. So we've got two graphs here, guys, showing you gibberellins, which is group A, and water, which is the group that was treated with water. And here we see on the x-axis, we see time over a period of time, and that was 16 days. And on the y-axis, we see the percentage of germination. Okay, so let's look at the questions that relate to this. I'm going to go on to the next page. Formulate a hypothesis for this investigation. And guys, if you remember, there's a big difference between the hypothesis and an aim. The hypothesis is often a statement of intent or what you plan to investigate in terms of to prove. An aim would indicate your intention in your experiment. So in this case, if we go back to the graph and we can analyze the graph, here we can formulate a hypothesis by looking at the information given in this graph. So here we see the effect of gibberellin and water in two groups. And I would look at this graph and say the following statement. Okay, I would say that plants treated with gibberellins have a higher percentage of germination as compared to plants treated with water. And you can clearly see that that's evident from the graph. Here we've got a group that has been treated with gibberellins, a plant hormone that stimulates germination. And here we've got a group of plants that were treated with water. So I would make a statement, and the statement would be without any personal pronouns, without having to say I think, I believe, or having to say prove, aim, or investigate. You've got to make a statement in terms of what would have been done by the scientists, by these learners before. They would have made a prediction, and that's more what a statement, a hypothesis. It's a prediction of what you. In this case, we've got the results. So the hypothesis needs to tie in with the information that is presented in their findings. So the hypothesis for this experiment would be plants treated with gibberellins show a higher percentage of germination as compared to plants treated with water. And that's exactly how you would get 2 marks for that question. Okay, so it's a 3 mark. So we said that plants treated with gibberellins show a higher percentage of germination when compared to plants treated with water. And for that, you would get 3 marks, and the 3 marks are obviously allocated for saying, identifying that it is a statement, and we're comparing the growth of germination with gibberellins, and we're comparing that with water, and we talk about the percentage of germination. So we've made a statement, we've compared the 2 variables that have been tested here, and the variables that the independent variables were the effect of gibberellins and the effect of water on the germination of plant, on the germination of these seeds. Let's move on to the next question. Using the information from the graph, so we've got to use the information from the graph, determine the percentage germination of hazelnut seeds on the 10th day of treatment with gibberellin. So we've got to go back to the graph, and that's on the previous page, guys. I'm going to go back to the graph. No, sorry, it's the next one. Okay, so here we are. So on the 10th day, so remember that when you ask a question like this, guys, remember, sometimes it gets very confusing. Identify the day, use your rulers, guys, project the line so that it cuts through the graphs, and the question it asks, determine the percentage of germination when plants were treated with gibberellins. So do not get confused. Remember that it's gibberellins, and in this case, it is graph A. So we've got to find that point, again, with using your rulers, guys. Extrapolate on the x-axis and try and use a straight line, unlike mine. And it was approximately 78, and don't forget the unit in this case, percentage. So it was 78% of plants 78% growth with the treatment of gibberellins. Let's move on to the next question. State 2 ways in which the validity of this investigation could have been improved. And often you find that the examiner wants to test your ability to analyze information, investigative skills. So if we go back to the method, and I intentionally did this when I was reading this, obviously because I knew exactly what the next question was. So the question was, analyze this to comment on how you would improve the design. If we look at this experiment here, guys, they mentioned that a sample of hazelnuts was divided into 2 groups, And often we could divide them into two groups, but it hasn't specified that there were two equal groups, and that's very important. We need to control those variables to make it a fair and a reliable experiment. So the two groups should have been divided. I would have said that they would have needed to be divided equally, and that would mean that you've got a group of 50 hazelnuts and you've got another group of 50 hazelnuts. That will make the experiment very fair. The next statement mentions that they were treated with a solution. It has not specified the amount. And for us to make that a true, a valid, and a fair experiment, we've got to treat them with the same amounts or a specific amount. And unlike an amount that you may take, you need to be specific about equal amounts or 50 mLs. So you've got to specify the amount of treatment that you've used. In this, you notice that they've added water as well and not mentioned the amount of water. So be clear, mention the amount of water. You got to be clear and scientific when you're planning an investigation. I'm sure you guys, when you cook, when you bake, the amounts are scientific. You measure specific amounts, and that's exactly what an experiment is about. The information should be very clear. Okay, I'm going to look at the last question, and then I'm going to see if you guys do we still have time to do the challenge question?
I think we should definitely get on to the challenge question, and then I can announce who the winner is. That's my favorite part of the show.
Okay, so the challenge question, guys, and I've thrown lots of clues to you. What process does the term oogenesis refer to? And I'm going to very quickly do this. So during the break, Indy was having a go at this word. She said oogenesis.
Oogenesis.
So it does sound cool, but oogenesis refers to, obviously we spoke about spermatogenesis, formation of sperm. So oogenesis would be a type of gametogenesis, or the formation of gametes, but it would be the formation of the female gametes, so the formation of eggs. When we look at the options that we're given to you, oogenesis, the secretion of estrogen, that's not oogenesis. Sorry for you. The production of haploid egg cells in the follicles of the ovary, very true. And the production of the male gametes, guys, is spermatogenesis. So the correct answer for this was B, and I'm sure Indy has picked up the winner, and she's going to announce that in a short while.
Gosh, it was so amazing having. I had a helper Marara in the studio the other day, and she helped me do a drum roll. So okay, maybe I should do my own drum roll. Wonder if they can hear me. Well done to Cordelia Dunjana. Well done for winning this awesome prize. You get CDs, you get DVDs, you get books, you get this awesome I'm a Mindset, a t-shirt. Welcome to the team, girl. And what you need to do is you need to, let me just double check, you need to email win@learnextra.co.za with your t-shirt size as well as your postal address. And just in case I said that name far too fast, I'm going to say it again. It's Cordelia Dunjana, and well done to you for answering the competition question correctly. And yeah, we think that's really, really awesome. So let me just quickly read. You guys have so many amazing. Rudolph just sent a picture of myself. I shall reshare it on the Facebook page. He said he is a huge fan. So thank you so much for tuning in. Guys, I've said it again. I'm going to say it one more time. Don't forget that these shows, the live shows, will be repeated this Saturday and the Sunday, paper 1, the Saturday, paper 2, this Sunday. We've also got exam schools that are coming up at the end of the month to help you through this process and this journey. We are with you from the beginning to the end. Don't forget that our Facebook page is for you guys to community, for all of us to share information and help each other out. Thank you so much, guys, for an awesome show. You guys rock. Have such a fabulous Friday. Goodbye.